All right, thank you. This is a bit of a follow-up from a talk earlier today by Steve Fry, who's the lead author of this talk. I'll present some of the work today. Basically, the problems are associated with waterborne pathogens, antimicrobial resistance genes, and veterinary antibiotics. Obviously, pathogens make us acutely ill. They're a public health concern. Bacteria can transfer genes encoding resistance to antibiotics from one bacterium to another. Bugs become more resistant to certain antimicrobials, making the treatment options more limited. Of course, the ultimate fear is the fear of the big resistant superbug, and that is a very real public health concern. And antimicrobials in organic waste supplied to land as fertilizers can potentially promulgate AMR in the broader environment. So these are the core issues, and they're core issues associated with manure applications that commonly occur on agricultural lands. We've heard a lot about that during this conference already. So in Canada, where we're doing this research, the surface, the manure application zone, the tile drain system, and groundwater are intimately coupled. And they're linked in many cases by large macropores that facilitate rapid transport of organic amendments to depth into groundwater and tile drains within minutes. And you can see here under this experiment here, which is biosolids we applied, that kind of contamination occurred within minutes into the tile and out through the drain into the broader environment where exposure risk can be manifested ecologically and through public exposure. So one of the practices that's promising for controlling or managing manure-derived pollutants to the to stream system, surface water, is controlled tile drainage. Tile drainage, again, is required in many parts of the world to drain the land that's otherwise too wet to grow crops. So it's an important requirement, especially in Canada, eastern Ontario, where wet conditions are certainly preclude early establishment of crops on the field and trafficking. But you don't need to drain water continuously, and managing and conserving it in the field is important for crop growth during the growing season. So the idea being is you put a structure on the header of a tile drain, and you can actually control that water, and ideally that water is used, nutrients and water are used by the crop to facilitate, to improve yields, and we see that occur during the growing season. So there's an agronomic angle to this, but there's also pulse mitigation of manure-derived contaminants that you can apply for certain situations if need be. And again, what we want to do here is if you're applying manure on fields and you can control drainage, you're controlling the mass losses of bugs and drugs into the surface water systems versus an open system that is able to move these things out in an efficient manner. So we have controlled drainage and a free drainage system. So we did some experiments, and Steve Fry talked a bit about that in another session. But the core question here is, can controlled drainage mitigate losses, mass losses of antibiotics, AMR genes, and Campylobacter, which is a pathogen, to surface water? So a study where we applied post-harvest, this is again after the crop was harvested in the fall under antecedent very medium wet conditions on barley stubble, monitoring 40 days after application of liquid swine manure, applied at 80 cubic meters per hectare. And the antimicrobials that were used in the feed for growth and health issues with the swine were chlorotetracycline and tylosin. And a byproduct of the manufacturing of those of the CTET was tetracycline, and that was also found in the processing as a byproduct. The field applied, we applied a water tracer, a rhodamine water tracer to the manure, creating this beautiful purple poo coming out of the applicator. But that was tracked with optical sensors to monitor its transport into the tiles. So Liquid chromatography, electrospray ionization, tandem mass spec approaches were used to characterize the veterinary antibiotics in water and a variety of transformation products. So we have the parent materials, tylosin, chlorotetracycline, and there's also tetracycline, but a variety of isomers and other transformation products were characterized. Pathogens, real-time quantitative PCR approaches, some assays that were developed to characterize, to quantify Campylobacter species in the water, as well as tetracycline-resistant genes in Campylobacter. An assay was developed to look at a Campylobacter-specific AMR gene, tetracycline-resistant gene, but it was found in much more than Campylobacter. There's a lot of cross horizontal transfer of these genes. So we used the tetracycline gene, and tetracycline gene was found in the system and in the manure as a model AMR gene to track in this study. And of course, we know there are many other clinically relevant AMR genes out there, but this one was used for this study. Again, the site is a macroporous clay loam field, flat plastic tile drains at a depth of around 80 centimeters. We had 3 plots that were under control drainage, 3 under free drainage. And we set the outflow depth, so before we applied manure, we set the gates at around 30 centimeters below surface, so when the water level in the field gets above that, it spills over and out into the ditch. So otherwise, if the water table is below that, there's no drainage from the control drainage. So hydrology, a suite of events, rain events occurred over the course of the experiment, initiating tile drainage for both controlled and free drainage. Essentially, the free drainage captured by volume around 80% of the incident precipitation, whereas control drain captured around 36% of the volume on a volume basis of the precipitation. So you're seeing that the control drainage is, in fact, managing water discharge from these systems after application of manure. Well, some core findings. So one thing that does happen when you apply manure to these leaky fields is you get an initial application-induced pulse into the tiles and into the groundwater. And for the control drainage, you see that there is nothing getting out in the system. That was application-induced, anything. So it was complete abatement of nutrients, drugs, bugs, everything. Whereas for the free drainage, of course, we're seeing here that we have a variety of analytes and drugs and nutrients, etc., moving out of the system as a result of direct preferential flow from the liquid application at time of application. Over the entire study period, we found Campylobacter was found in tile and groundwater pre-application, but there was no antimicrobial resistant genes found until after application. It makes sense. So you can see that here. So Campylobacter is in the system. Birds come on, what have you. There's DNA in the system. But the antimicrobial resistance genes only appeared after application, and that is indicating very clearly that the source was the swine manure. And controlled tile drainage loading was less than the free drainage loading during the study period for both of those targets. However, controlled drain veterinary antibiotic loads were indifferent from free drainage loads. So they're kind of counterintuitive. So we would expect that the antimicrobials would behave similarly to the bugs, but they don't. So just to kind of reinforce that with regard to a suite of nutrients and fecal indicator bacteria, for the nutrients, we see higher concentrations in control drain effluent, but no significant differences in loads. And for the fecal indicator bacteria, we see higher loads associated with free drainage and no significant differences in concentrations. So for shallow groundwater, there's no detections of veterinary antibiotics pre-application and no significant differences between control drain and free drain in shallow groundwater. But for Campylobacter and TET-O, post-application concentrations in groundwater were significantly higher than pre-application concentrations. So there's some interesting stuff going on here. So to summarize some of these plot experiments, manure-based veterinary antibiotics, the rhodamine tracer, nutrients behave similarly with respect to tile drainage movement management. There were no significant differences among controlled drainage and free drainage over the entire study period. The bugs and the AMR genes behaved differently, however. Controlled drain loads were less than free drain loads. And controlled drainage was, of course, very successful at fully abating application-induced tile contamination when the manure was applied. So the reasons between the colloidal versus the solutes, we're not sure why they're behaving that way, but it could be something due to those very attributes. One's colloidal, one's solute, and how they behave in relation to transport in these macropore soils remains something of a research concern for us right now that we're pursuing. But under different weather conditions, we may have had different results too. So we have to always be cognizant of that. So if we scale this up, we see some attributes over managing over a short time period, managing manure-derived pollutants for that purpose. But if we're managing controlled drainage for agronomic purposes over the growing season, and here we have a watershed where we have entirely under controlled drainage and a watershed entirely under free drainage, and if we look at how bacterial endpoints, other viruses, other kinds of microbiological targets behave over the growing season, we see that we can actually reduce significantly Archobacter, Salmonella, Campylobacter, and a variety of microbial markers here that are indicative of source. So these kind of microbial source tracking endpoints, which is livestock, Bacteroides marker, or a ruminant here, and human markers were significantly reduced in the context of occurrence. So over the big scale, we do see that this practice does have potential, of course, to mitigate over a long time period a variety of important pathogens from getting into the surface water system. So while we have some kind of interesting counterintuitive results at that plot scale over that time period for those macropore soils, the core idea of controlled drainage during those manure application events where you don't want any of that getting out into the surface water is time. So you want to manage. So we may be here, you're seeing like the dissipation of fecal indicator bacteria over time. Keeping that in the field as long as possible, of course, is going to help reduce exposure risks downstream. And so we feel that despite some of the findings with regard to veterinary antibiotics, this practice over a course of a pulse experiment is certainly prudent if you're applying a lot of manure on leaky soils. So thank you.
Thank you. Questions?
You proposed that the difference between the chemicals and the bugs is controlled by colloidal phenomena for the bugs, eh?
Maybe.
Yeah. But then the microorganisms themselves can also behave quite different in terms of colloidal interactions with solids. So do you have any idea about the characteristics of all these different microorganisms and how they could control their colloidal transport in such a system?
Yeah, I don't have a good answer for that. I think it's a very complicated question, and I mean, it's something that's been done on looking at column experiments, and there's been a lot of very controlled research experiments looking at cryptosporidium transport through macropores and so on. And there's, in these natural systems, it's unclear. It could also be related to how the water table interacts with that manure zone, somehow preferentially mobilizing these colloidal clumps getting down into the drains. It's unclear at this time. I think some modeling work, and Steve had presented earlier on that, might help us elucidate some of that, but it's not clear at this time.
So some of these indicator microorganisms, they are known to have flagella and these type of things, and they help,
like active transport,
and they help, and they determine their stickiness to all kinds of things. So that might be also a thing to take into consideration.
Yeah. So the protozoa, we know that these active transport is certainly a part of that equation, but I think that's probably, to be just intuitively, I think it's probably rescinded to just a contact or some sort of macropore interaction that we're not understanding fully yet.